Hello, thank you for joining us today as we discuss InhibiScreen, a kinase inhibitor assay, in greater technical detail. My name is Bethany Bell. I am a Senior Product Manager at ALPCO, responsible for managing InhibiScreen, and am available at any time should you have further questions. InhibiScreen is a cell-based assay that we specifically designed and developed for pharmaceutical companies, biotech companies, and their functional service providers for the evaluation of efficacy of inhibitors, specifically of lipid kinases, during several different phases of drug development. Those phases of drug development where this product can be utilized are lead discovery, lead optimization, and preclinical development. The way that this assay functions is based on the suppression of the in vitro stimulation of basophils in the presence of inhibitors of molecular targets such as PI3 kinase gamma and PI3 kinase delta. Suppression is measured by flow cytometric analysis of externalized CD63 antigen expression on the basophil cell surface after degranulation. When you receive the InhibiScreen kinase inhibitor assay, the following components are part of the package. The first set of components are for the stimulation and staining portion of the protocol. Some of these are provided in a lyophilized format, which need to be reconstituted prior to use. So there's a stimulation buffer as well as stimulation controls. The controls are an antibody directed against the IgE receptor. This is reflective of the delta isoform of PI3 kinase, as well as the FMLP stimulation control, which functions through the gamma isoform of PI3 kinase. There are also staining reagents which are ready to use, which include the CCR3, which is PE conjugated, and CD63, which is FITC conjugated. For the second half of the protocol, lysing is required, and the components within this protocol are the lysing reagent, which contains a fixative. This is provided as a 10x concentrate, which needs to be diluted to 1x prior to use, as well as a ready-to-use wash buffer. Some additional information about the stimulation controls. The two stimulation controls provided with the kit allow for the study of specific inhibitors of the class 1A and 1B PI3 kinase isoforms, delta or gamma. So as mentioned on the previous slide, anti-IgE is signaling through the delta isoform, and this can be used as a stimulation control when studying inhibitors of PI3 kinase delta, as well as BTK and SYK. The second stimulation control, FMLP, signals through the gamma pathway, which can be used as a stimulation control when studying inhibitors specifically of this isoform. The staining reagents that are provided are directed against CCR3 and CD63. CCR3 is expressed on basophils, eosinophils, Th2 cells, and mast cells. So when you're looking at a dot plot generated after flow cytometric analysis, this is the kind of thing that you're going to see when you're staining for CCR3. You have many different populations, as demonstrated here and here, which are illuminated in the presence of CCR3. What you're going to look for is those cells with a low side scatter profile and a high CCR3 expression. So this population right here is representative of basophils. CD63 is a gold standard marker of basophil activation, which is linked directly to degranulation. So on this dot plot, what we're showing here again is this high CCR3 population, which has low side scatter. So we've taken this population, investigating it further for whether we have CD63 expression, and high CD63 expression is indicating those basophils which are activated in the presence of a positive control. This assay provides flexible testing scenarios, whether you are studying delta, gamma inhibitors of PI3 kinases, BTK, or SYK inhibitors. The following testing scenarios would be used. For delta, for example, you would start with whole blood, which is either collected in the presence of EDTA or heparin. You would pre-treat with inhibitors at different different concentrations or a compound library of inhibitors. You would stimulate with the anti-IgE antibody, which functions through the delta isoform pathway of PI3 kinase, in addition to stimulation buffer and staining reagent. And this would allow you to visualize the ability of your inhibitors that you're screening and the way that they're actually inhibiting CD63 expression on these cells. And I'll get into some data to really explain to you how this is working. The same can be used for the gamma inhibitor screening. And again, when I mention assay flexibility, I'm referring to you have the ability to look at many compounds. So you can take a compound library and look at the ability of these compounds to inhibit either the delta or gamma isoform of PI3 kinase. The other scenario can be 10-point dose response curves or 25-point dose response curves. So you can really hammer in, hone in on what the IC50 of these compounds is, as well as do the upfront screening to determine which compounds are the best candidates to proceed forward with for testing. So a little bit of additional detail around the protocol for InhibiScreen. During the first step, you want to make sure that you reconstitute the stimulation controls and buffer, and you dilute the lysing reagent to working strength. So prior to stimulating the cells, you want to pre-incubate with the inhibitors. So either this is an inhibitor compound library or it's a dose response titration. And here we're recommending roughly 60 minutes at 37C. Following this pre-treatment with inhibitors, you will be stimulating and staining cells using a ratio of stimulation buffers, control, staining reagent, and blood. And following the addition of those components, you will mix gently and incubate for 15 minutes at 37. Once this stimulation and staining step is completed, you'll add 2 mL of pre-warmed 1X lysing solution. After mixing and incubating at room temperature, you'll centrifuge, remove the supernatant, resuspend, and if you are using a 96 well plate format, you will need to perform a second washing step. Lastly, during the data acquisition period, if you've found that you've run out of time for the day, it's perfectly acceptable to cover your tubes or your plate and store them at 2 to 8 for up to 96 hours prior to analyzing them on the flow cytometer, or you can analyze immediately. So the type of data that you will see when you're using this assay is demonstrated here. So for example, when activating with an anti-IgE antibody, what you're doing is you're activating the PI3 kinase pathway via the delta isoform. And in this experiment, we've looked at Cal-101, which is a known inhibitor of PI3 kinase delta. And what we've done here is we've started with making sure that we're looking specifically at basophils by graphing or taking forward the group that's low side scatter, which is shown here in the top left quadrant, and high CCR3 expression. So we've decided to gate this population as the basophils, and this is what we're focusing on for the remainder of the experiment. So when you take this population of basophils, you stimulate with the anti-IgE antibody, and you look at the ability of that to activate cells, which is looked at through expression of CD63, you identify this population as being activated. In the presence of 100 nanomolar of Cal-101, you can see that while the majority of this population is activated in the presence of the positive control, Cal-101 has the ability to shift this population down and demonstrates that this concentration of Cal-101 is roughly inhibiting about 50% of this activation. There are many examples in the literature amongst pharmaceutical and biotech companies where basophil activation tests have been incorporated as screening studies. And we have written a blog series which describes how basophil activation testing has been used in the development of several compounds, and specifically in the development of Gilead's Cal-101, which is the only PI3 kinase delta inhibitor to be approved by the FDA today. So why should you incorporate InhibiScreen into your current workflow? ALPCO's InhibiScreen Kinase Assay was developed specifically to help you fail those compounds faster which are ineffective, or those compounds which are targeting too many isoforms of PI3 kinase and could potentially result in unwanted side effects. So while it's been specifically validated for use with inhibitors of the delta and gamma PI3 kinase isoforms, we've also demonstrated its utility in BTK and SYK inhibitor development. And if this is something you're interested in seeing more of, I can certainly provide that to you. This is a phenotypically relevant testing paradigm. So if you're in early clinical study phases, you're using whole blood from donors. So if you want to look at the ability of these inhibitors to truly inhibit its target in donor blood, that's what this assay has been developed for. There's no special cells or equipment required. It's a standard flow cytometer fitted with a blue laser. It's a very flexible assay format whereby you can look at a 96 well plate format, you can do individual tubes, you can screen compounds, you can specifically zero in on IC50s. And the testing concept was utilized during the development of Cal 101 by Gilead, as mentioned. Additionally, data is reproducible when samples are acquired immediately following staining at 0 hours and even up to 96 hours post-staining. Here's my contact information. If you have any other questions, you're welcome to reach out to me at any time. I am happy to arrange evaluation kits for those folks who are interested in trying to understand better whether this assay will be beneficial to them during drug development. Thank you for your time today, and we look forward to working with you.